Hi, everybody. Today we're going to look at a tiny little topic for now, and then we'll turn it into a bigger topic later. So this is the third in our series of biochemistry notes. We're going to just take a quick peek at nucleic acids. And the reason why this is a quick peek is because the nucleic acids will come up again when we get to genetics a little later on, and then we're going to go all in with nucleic acids. So we're not going to do a lot right here, but I just want to kind of remind you of a few things about nucleic acids. The first thing is that the nucleic acids are DNA and RNA, and the nucleotide is the subunit or monomer of nucleic acids. And nucleotides consist of three things. They consist of a phosphate, a sugar, and a base. And that phosphate, it's the same phosphate you're going to see popping up in ATP. It's the same phosphate that's going to be part of a phospholipid, and then it's going to show up here. So a phosphate is actually an inorganic component. And again, it's one of those functional groups, I think, that we'll talk about when we talk face-to-face. The sugar is usually referred to as a pentose because it's a 5-carbon sugar. In DNA, that sugar is deoxyribose. In RNA, that sugar is ribose. And then the bases are nitrogen-containing or nitrogenous, and they're adenine, guanine, thymine, cytosine. Now, where we go a little bit more in depth here that we don't in baby bio is that some of these bases, the purines, which are adenine and guanine, consist of 2 carbon rings, and then thymine and cytosine, and then in RNA you have uracil, are what we refer to as the pyrimidines, and these have a single ring of carbon. So you're always bonding a purine to a pyrimidine when you're bonding them together in the rungs of the DNA ladder. The reason for that is you have something with 2 rings bonded with something with 1 ring so that the DNA is even all the way across, right? Like that. You can't have 2 purines bonding together because then you'd have a section that's 3 carbon rings and then a section that's 1. And so the DNA molecule would be kind of curvy if you did that. The DNA molecule itself, while it twists, it's a straight up and down molecule. And so you're always binding a purine to a pyrimidine. Specifically, the complementary base pairing rule says adenine binds to thymine in DNA, uracil in RNA, and then guanine binds to cytosine. Okay, so A with T in DNA, A with U in RNA, and G with C, always. And when we're talking about that bonding, remember that it's hydrogen bonds that are holding those purines and pyrimidines together. So it's more of an attractive force. And the reason why you're binding those specifically together is because of the number of hydrogen bonds that each wants to make. So A binds with T or U because they want to make the same number of hydrogen bonds. G binds with C because they want to make the same number of hydrogen bonds. It's the sequence on the DNA molecule, right, that's going to be written into the RNA molecule, and then that RNA gets translated to create proteins. So these nucleic acids, they're not nutritive. We don't think of them as being for metabolic purposes. We think of them instead as being for making proteins. They're information storage molecules. And also we can track sequential changes in the DNA between species to give us an evolutionary clock. So we can actually use that as well. So a little piece of information there about nucleic acids. All right, so here are the nitrogenous bases, okay? And I'm sorry, my face is kind of, there we go, in the way. I'll take my face out. The nitrogenous bases are here. Here are the pyrimidines. There's cytosine, thymine, and uracil, and then here's adenine and guanine. And again, you're binding A to G or U, A to T or U, and then G to C. And that way you're always binding something with 2 carbon rings to something with 1 carbon ring for evenness. And then here are those pentose sugars. Here's deoxyribose, and here's ribose, and there's the difference. This is a hydrogen here. That's why it's deoxygenated. Okay. So like I said, we'll get more into DNA a little later on, and we're going to do a DNA modeling activity together. So this is just giving you a quick overview of the components that you're going to see as part of DNA. And finally, we have hybrid biochemicals. This is a name that I've made up. These are chemicals that are combinations of maybe 2 different families of biochemicals, or they're combinations of an organic family of biochemicals and an inorganic component, right? So I just want to mention that these are out here. We're going to see these popping up as parts of components of cells. So glycoproteins, like protein and a carbohydrate. So here's a glycoprotein. That's what you're seeing. There's this protein component embedded within this membrane, and it's got a carbohydrate complex on its surface. You also see lipoproteins. Cholesterol packaged in protein by the liver is referred to as an LDL or an HDL, right? The LDL is the low density lipoprotein. That's the bad cholesterol that floats around in your blood and clumps together. And then there's the HDL, which does not float, it sinks. It's the high density lipoprotein, so it sinks and gets carried away from your body. And then again, you're also seeing the hybrid biochemical, the phospholipid, right? These are the little lipid tails that are part of that phospholipid in the membrane, and they're attached to an inorganic phosphate. So these are what I'm calling the hybrids of the biochemical families. But anyway, that gives you some idea if they pop up again. You're going to see some things like collagen. That's an example of a hybrid biochemical as well, and so on. These are going to pop up in cells a little later on. So important that we know.